Good day, this is Professor Will, CBMD PhD. Today is Thursday, September 24th, 2020. It is 2:35 and 51 seconds p.m. Eastern Standard Time. And this is a continuation of my microbiological lecture, so let's hop to it here. It is in information processing that the differences between eubacterial and archaeal cell designs appear in their full glory. Of the three main information processing systems, translation, transcription, and genome replication, translation exhibits the most universality in its componentry, the most homology between archaea and eubacteria and eukaryotes. Ribosomal RNAs, elongation factors, ribosomal proteins, aminoacyl tRNA synthetases are all basically universal. However, among these universal protein components, a clear distinction exists between the eubacterial and archaeal versions. The distinction is characteristically so blatant that it tends to stand out even upon gross inspection of a sequence alignment. For example, the eubacterial versions of a given sequence often contain a moderate to large blocks of amino acids, located terminally or in the interior of the molecule, not seen in the archaeal versions, and vice versa. In addition, for those columns in an alignment where a composition is highly conserved, one often sees that the characteristic eubacterial composition differs from that characteristic of the archaea. This nearly qualitative distinction between the archaeal and eubacterial versions of a sequence, this difference in the genera between the two has been called the canonical phylogenetic pattern. Differences this extreme are never encountered among the various eubacterial taxa, or among different archaeal taxa, although in each case there have been 3 billion years or so, most of this planet's history, in which such differences could have arisen. When eubacterial and archaeal sequences show these differences in genera, their eukaryotic counterparts are almost always of the archaeal genera. A minority of ribosomal proteins, however, are not universally distributed. A relatively small cadre are characteristic of and found only in eubacteria, while a somewhat larger set is common and confined to the archaea and eukaryotes, with yet a few others being found only in eukaryotic ribosomes. Overall, though, translation gives the impression of a system that existed in basically modern form at the root of a phylogenetic tree. Good day, this is Professor Will, CBMD PhD on microbiology, bioinformatics, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.